Brian, could you tell me what's one difference between these two viruses that I drew?
One has a tail, the leg.
Mm-hmm. So one has a tail and the other one is kind of more of like a sphere-looking thing. Doesn't have a tail. Okay. Adriana, what's another difference between these two?
The envelope.
The envelope. Mm-hmm. Yep. Mm-hmm. All right, so this virus on the right has that lipid bilayer envelope, which, J.U., how did that benefit this type of virus? What does that allow it to do?
It allows it to kind of like seep into
Yeah.
the cell.
Mm-hmm. What do you mean by seep into the cell? Like, what happens during that?
Binds
Mm-hmm. It binds, it fuses those phospholipids of the envelope. They fuse with the phospholipid of the cell, and then it can, like, dump all of the material inside of there into this cell. Yeah, Wenji?
There's a specific name for like that bacteria, like that virus, because the tail one is called phage.
That's a good question. So he asked, is there a specific name for this virus? I gave like 3 different examples of it. Yes and no. It depends what type of genome they have. So if this is a virus with a RNA genome, you're typically looking at a retrovirus. But there's all these other types like lambda viruses, and there's like almost species, like classes of virus. But the 2 categories here are with and without envelope was kind of the categories that I was making here. But you don't need to know those other names of virus categories. Okay. Hopefully, everybody had a chance to watch Mr. Anderson last night. He talked about viral infection, right? And how he gave a little bit more detail about what I described here, right? How when a virus injects or fuses with the host and dumps its nucleic acid into the host, it then uses that cell machinery to make copies of its nucleic acid, transcribes it, translates it, so that it can make proteins. And with the genome copies and the proteins, they can form new viruses. Okay. So the third objective that I had for this set of notes was actually what can we learn from viruses and why are they useful? Now, that second question might seem a little bit strange because usually viruses are brought up in like a negative context because of the infectious diseases that they cause, but there's actually a lot that they can teach us about how organisms work and the machinery that allows that. So the last thing we're going to talk about is that third objective. I'm going to add a little bit more space here. Okay. So this third objective, again, is... Oh, no. Wait.
What?
Wait, I'm telling you, the room is cursed. I don't know what's going on. We can do it. Deep breaths.
Wow. Wow.
And we're good. So, third objective. What can we learn from viruses? Why are they useful? Okay. And there's 4 major answers to this question. The first one, the most kind of immediate one that comes up is that there's a medical need, right? We see this currently, we've seen it before, that there is a medical need to understand different viruses. Although viruses share a lot in common, each virus kind of brings something different to the table in terms of how it infects, what proteins are involved. And so when we understand this, this helps create prevention, treatment, and cures. So one of the big areas of viral research, of virology, is around HIV, which we're going to talk about. I think I got embarrassed when I started sharing my past experiences with this projector. That's probably what happened.
You got jealous.
Okay, so what we're going to talk about later in this unit. All right, this is ridiculous. Let's pause for a sec.